Okay, so I think I finally figured out a way to successfully record these lectures. If you can't make it or you want to revisit the lectures for studying, you'll have that available to you from here on out. Okay, so today we're going to talk about a little bit more about memory. We talked about it a bit last week. And we'll also be talking about associative processes. So associative processes are things like learning. So this is all of that, CS, US, all that stuff, classical conditioning, operant conditioning. A lot of people find that stuff kind of dry. I agree. It's a little dry, associative processes. But memory is very fun, so we're leading with the fun part. The thing about associative processes, though, very important to understand, to know how learning works in that way. Very important contributions in that field. So we have to talk about it, even if it is a little dry. All right. So let's start with memory, part 2. And we'll just do a little bit of revisiting here with the stages of memory. We talked about this last week. But an important thing to remember about memory is that it is a process, and it is not necessarily stable, right? Memory can change. It is liable to change. And there are 3 stages of processing in memory, which you can see at the bottom here. We have our encoding, our acquisition of information, sometimes known as learning, or referred to as learning. Our storage, our retention of information. If it's unsuccessful, that can be known as forgetting. And the retrieval, the recovery of information, or remembering. Right. And you can see that shown here with time, encoding, storage, retrieval. We also ended off last week talking about how storage might not be just one kind of phase. There can be different phases in storage. And another word for that storage is consolidation, so the consolidation of memories, right? We talked about how there might be 2 stages of this consolidation, or this storage. And we say that because we find that when protein synthesis is disrupted, it can interfere with memories immediately, but not after a period of time has passed. And then a disruption in the second phase of this storage has to be either a lesion, so damage to brain tissue, or a disruption of synaptic transmission. So experimentally, we found these 2 differences, and that's why there's some people theorize that there are 2 stages of consolidation. Also known, one of these stages, as reconsolidation. So reconsolidation is when memories are being reconstructed. And when that happens, they are liable. Similar to when they are being consolidated. Okay? And this concept of reconsolidation has actually been demonstrated in many different species. And you can think about it like memory updating. When you're recalling something, the memory gets reconsolidated. So it changes over time. Right. I will say, though, that not all evidence supports reconsolidation as a super distinct phase. It's not really clear-cut. Still trying to figure it out. But important to know. And your textbook discusses this as well, in this way. Okay, so some evidence for reconsolidation. Here's an experiment that was testing reconsolidation. Okay, we're going to talk about shocking rats. So at the top there in A, if you give a rat a shock and pair it with a bright light, okay, 24 hours later, if you just show the rat the bright light, they'll freeze because they're expecting to get shocked, right? Now let's say in B, you have that bright light, you have that shock, pairing those two stimuli, right? But right after that, you inject anisomycin. And this blocks that protein synthesis, okay? 24 hours later, if you show them the bright light, no freezing happens. Okay, interesting. So you've kind of blocked that association. You've blocked that memory to pair bright light with shock. However, in C, if you do that same conditioning, you wait 3 to 4 hours, you inject, you still see that freezing. So this is, the authors of this paper here are theorizing that there are 2 different stages of that storage, right? Time has passed, but the amount of time matters. So the memory formation here wasn't blocked, right? So that's some evidence to support this reconsolidation and consolidation as being separate phases in storage. Okay, you can look at this a little bit more further. So in D, again, still pairing a shock with a bright light. 24 hours later, you test them. They're gonna freeze. But okay, right after testing, you give the anisomycin. And then later, 24 hours later, you can see no freezing, which is interesting. However, in E, have that conditioning, do the testing, and then wait again for a period of 3 to 4 hours and do the injection, you see freezing. So the amount of time here is important. And this can actually have some pretty interesting therapeutic benefits in human beings. So what do I mean by that? Well, there are actually applications of this kind of work. So something kind of a bit more famous from this work is being able to alter fear memories, okay, with propranolol. Propranolol. Here's the paper here. Okay, so let's say you have a fear, you have a deep fear of spiders, right? Anytime you see a spider, whew, freaks you out. What you could do is get this kind of therapy where they'll bring you into a room with a spider. They'll ask you to get close to it. Maybe they'll ask you to touch it, if you can. And then right after that, they give you this drug. Even though you've had this fear memory several times, this has happened several times, right? You're not getting the drug the first time you were scared. It actually lowers the return of the fear over time. Right? So if we go back to that example with the rat, even though the rat's been conditioned and they've been tested, because they get the injection of the drug, we're looking at D, right after that testing happened, it blocks that. It blocks that. So same kind of thing here. Pretty interesting. Cool application of this kind of work. Okay, so now we're going to move on to some different classifications of memory. When you study memory, when you learn about memory, people are always going to talk about different taxonomies of memory, different ways of splitting up memory. The most basic one you might hear about is short-term and long-term memory, right? Those terms aren't really usually used in a scientific context for memory, but you can think of working memory as a short-term kind of memory. It's memory that's online. I'm using working memory right now talking to you all. You're using your working memory listening and trying to apply it to what you read, what you've previously learned. So it's maintaining information in a short-term store that can be used in cognitive processes. Yeah, so working memory is active. It's an active process. You're holding information online. You can also think of it as a single trial memory in the context of a task. Having to remember a number, you're using your working memory. And talking day to day, that also uses working memory too. A very popular task for studying working memory in humans is something called the N-back task. So generally you're presented something like a number or a letter, and then you have to remember, well, you'll be presented several stimuli. And you have to remember how far back you saw that first stimulus. So here's the N4 task. So N being any number, can be an N2 task, N3 task, N4. The number just means how many presentations of stimuli you have to remember back until they match, right? So you can see here you're given A and then several other letters, but you have to remember on the fourth one, does it match the fourth one back? This task gets harder the more you have to remember back, right? It's easier if you only have to remember 2, but you're holding all these items in your working memory. Okay, A, H, H, A, correct, right? So it's an active process, right? People with Alzheimer's and other neurodegenerative disorders, this is a difficult task. So even though they can read the letters, they can read the numbers, it's really hard to remember back a few tasks. We also see this in patients with prefrontal cortex damage who might actually have no issues with remembering things in their past. But they do have an issue. They have a disruption in performance on working memory tasks. So the different types of memory are also associated with potentially different systems in the brain, different places in the brain. There are different memory systems, right? And working memory is one of those. Okay. All right. That's one way you can study working memory in humans. In animals, we often use something called a delayed match to sample. Of course, humans can also do delayed match to sample, but this is a pretty common one in a lot of animal work. All right, so first, a sample stimulus is presented to the animal. I'll show you what this looks like in a second. Then that sample stimulus goes away, and then you give the animal a set choice of stimuli that are presented. I forgot to mention, this is usually on a touch screen. So you can train primates to use touch screens with their fingers, right? You can also train birds to peck at touch screens. Okay, so sample stimulus presented, sample stimulus removed, you're given a choice, and they have to match that choice to the sample. Now the delay between the sample and the stimulus presentation tests working memory. Okay, I'll show you an example of what this actually looks like. So this is a brown-headed cowbird doing a delayed match to sample task here at Western. I worked with these guys in my master's, And later on, I'll actually give you a little bit of a research talk on some stuff that I found during my master's with these cowbirds. But for now, okay, delayed match to sample. So we're going to watch a cowbird peck these screens, and this happens here, or it did, at Western. Okay. So bird enters cage. There's a little food hopper down here that rewards are going to come up through. And here's the touch screen. To start a trial of the bird first, while he's picking up the leftover seeds, of course. To start the trial, he pecks this. Green circle. And then he'll be given a sample. There's the sample. Gotta peck that. Okay, I'm continuing on. I have to peck the blue one to be given my choices. But he messed up. Didn't do it fast enough, right? Gotta pay attention. Start over. Peck. Sample. Peck. Come on. Choice. Okay, got it right. Reward. Why is that correct? Let's keep watching and see if someone can figure out why that one is correct. Okay? Sample. Choice. Correct. Nice. Delicious seed. Sample. Choice. Correct. Nice. This guy's doing pretty well. We'll wait until he gets one wrong. When it's wrong, they get lights out. Still correct. Okay. It's pretty good. I know he gets one wrong in here somewhere. It's not that long in the video, right? Yeah. Ah, wrong. Lights out. No reward. All right. Can anyone tell what the correct choice is from that? Yeah?
Is it just like the placement of where the last one is?
It's the location. Exactly. So this was kind of testing their spatial memory, right? Can he remember in his working memory the location of the correct choice? You can do all sorts of delayed match to sample tasks. You could do it for size. You could do it for color, move things around, right? And I'll talk a bit about that later on. But yeah, delayed match to sample, very frequently used in working memory research with animals. Oh, yeah. All right, but there's something, I think important to say about touchscreen work, and that is the ecological validity of tasks. So, are touchscreen tasks ecologically valid, do you guys think? I want to say something about that. What I mean is this something that's happening in nature? No, right? There are just touchscreens hanging out in nature. Although there are some researchers who've tried to set up touchscreens in nature to do these kinds of experiments without having to capture birds, but this doesn't just happen, right? The thing about a touchscreen task, though, is that you can get lots of trials, you get lots of data pretty quickly, and it's automated, right? So you can still ask really interesting questions using touchscreen tasks, but we do need to think about the ecological validity. Is an animal in nature just going out, pecking things, and then getting rewards? Not exactly, right? And this is something that comes up a lot in this area, is the ecological validity of tasks. What are you actually measuring? as well. What are you measuring? What aspect of this animal's life history is being tested here? So how should we design tasks? You know? There's always a trade-off, of course. So with tasks like these, you get lots of data. But yeah, there aren't just touch screens hanging out in the wild. Radial arm maze is another task that's pretty common in this kind of work. And this was actually designed with the rat's natural history in mind. Okay, so they were trying to make a more ecologically valid task to assess working memory in rats than just pressing on a lever, right? Or touching a screen. Here, at least the animal has to move through the environment to get rewards, to solve tasks, right? So basically, you have your rat in the middle here, and you have these different arms. And there could be food rewarded, right, at different arms. Right, see, there's a food reward here, none here, etc. And there's all sorts of things you can measure in a radial arm maze to study working memory. And here we can watch a nice little video about an 8-arm radial maze.
Two maze engineers. Maze basics, 8-arm radial maze. The 8-arm radial maze has 8 arms connected to a center. A mouse sits in the center and tries to find food by exploring the arms of the maze. After each choice, the mouse must return to the center and wait. Behavioral parameters include the number of arms the animal explores before finding food, the number of different arms the animal explores before finding the food, the order they choose the arms, the first arm they choose, how fast they get the food, how far they travel, how long they pause at the center each time, how long of a forced pause between choices affect memory, and how many trials it takes them to learn where the food is.
All right, just want to pause here. So these are all sorts of different dependent measures you can get from this maze. So when someone's doing an experiment with this kind of maze, they might not be looking at all of these different factors. But generally, the number of arms they check before finding the food and revisits are pretty important. If they go back to an arm that they've already been to, that's considered an error. So it's not important necessarily to know all these different dependent measures about a radial arm maze. It's just important to know what the task is and kind of what kind of questions you can ask with this maze. Okay, continuing.
Each parameter can tell us about different processes that contribute to the overall ability of mice to navigate the maze. A common area of study with the 8-arm radial maze is reference memory and working memory. Reference memory refers to the knowledge for a certain situation that remains constant over time. Working memory is the ability to manipulate or use the memories being held. Consider this simple task designed to test reference memory. The mouse waits for the doors to open before the 50th trial. It has already done 49 trials in which the same 4 arms were baited every time. If the mouse correctly goes into one of the baited arms, it can eat the treat. Access to previously chosen arms is denied to prevent working memory errors. If the mouse chooses an arm that has never been baited, it commits a reference memory error. The trial ends when the mouse gets all the food and the maximum number of reference memory errors per trial is 4. The following sample task is designed to test working memory. All arms are baited and the mouse waits for the doors to open. With the first choice, the mouse always gets the food. With the second choice, the mouse may correctly choose a new arm and receive more food. However, if the mouse returns to an arm it already chose, it commits a working memory error and doesn't receive a treat. The trial ends when the mouse gets all the food and the number of working memory errors per trial is limited.
Right, so working memory error, revisiting an arm you've already been to, right? That's something that they should be holding in their working memory.
Let's By manipulating task parameters, the memory load can be increased and decreased, which is important because often memory impairments are subtle and may require certain conditions to manifest. Using both types of tasks and variations of them is important because deficits in working memory and reference memory can be disassociated. By manipulating the affective states of the animals or using rodent models of diseases, researchers can use the 8-arm radial maze to study interactions between reference and working memory and other brain networks. The 8-arm radial maze is a powerful and extensively used tool due to the minimal stress placed on the animals and the ability to study different cognitive processes using a single type of maze. MazeEngineers.com.
All right. Maybe we should check out MazeEngineers.com. I really want to. No, that's okay. Okay. So, one way we can test working memory in rodents. And also reference memory, which we will get to. So yeah, this is just a graph showing how arm revisits mean you'll have more working memory deficits. Okay, so why have working memory? Well, it's important for updating information to a changing environment, right? Things change, you need to be able to be flexible. To be able to react to that changing information. Of course, we often think about memory in terms of what we know with humans, right? And it can be difficult to test a lot of, things in animal cognition in general, because we can't just ask animals things. We have to be a bit more clever about how we design our tasks and how we ask those questions. But we do know from work with humans, right? We all have an intrinsic nature of what working memory is. We know that it allows us to think about things that are not actually there, right? And there is a theory that this capacity for greater working memory is associated with our larger prefrontal cortex. And that things like designing tools and using language may require you to maintain and update information more regularly. But non-human animals without a large, densely packed prefrontal cortex also have working memory. They do. Okay, reference memory. That was briefly discussed in that little video there. And some people would call long-term memory reference memory. So that's the process of using information in that long-term store. Okay? And again, this is a different memory system. So it functions independently from other processes like working memory. And we often talk about these different processes, these different memory systems in the context of HM. Does anyone know who HM is? Henry Molaison. We actually know his name now because he died. He was released. Yeah. Sorry, what was it? That's right. So he had part of his brain removed, and it gave him some pretty severe impairments to one of his memory systems, but not to another. So, for example, he improved on tasks, a memory, or what's it called? It's like a tracing task. He improved with that over time. but he had no memory of it happening. It's a task that initially, when anyone does it, pretty difficult, but over time, you get better and you improve. But he had no memory of actually doing this task, even though he was clearly improving. Right. So different memory systems. We have different memory systems. Pretty famous case of HM. I'm sure he'll come up in your other courses. But yeah, important to know different memory systems. Okay. So in the realm of reference memory, you can break it down into non-declarative and declarative memory. So non-declarative meaning it does not depend on explicit knowledge, it is implicit. And then declarative memory is explicit knowledge.